Hello guys, so today we are going to discuss about cloning vectors and competent host. Okay, so today we are going to discuss how many topics? Two topics. One is cloning vector and competent host, right? So it's a very interesting chapter, right? Now, cloning vectors. We have already learned what is a vector. Vector is like a vehicle. It is like a vehicle which is carrying the foreign DNA or the gene of interest from one place into the host. Okay, so you can see what is a vector. Vector serves as a vehicle. Okay, so what they are carrying? They are carrying foreign DNA sequence, which is the gene of interest, like pest resistant gene is a gene of interest, into a host cell, into some plant it is carried. Okay, so that is the role of what? This vector. And usually the vectors what we used is plasmids, that is present in bacteria, as well as there is a virus named as what? Bacteriophages. The virus which attacks bacteria, bacteriophages virus, that can also be used as a vector. So normally what are the two vectors which we are using? One is the plasmid, that is present in bacteria. Extra chromosomal DNA, circular DNA we have learned, isn't it? So that is plasmid, and another one is what? The bacteriophage, right? Now, this is an example which is given in your text of a vector. What is the vector here? It is a plasmid. This is a vector, and this vector is PBR322. Name of the vector is what? PBR322. Okay, so it is present in E. coli. E. coli, you know, isn't it? So in the E. coli, in special E. coli cells, you can find this PBR322 vector is present, plasmid is present, and they are playing a role of a vector. Means it is carrying the gene of interest from one place to the host, right? Yeah, so that is the role of what? This PBR322. Now, when you see what are the characteristic features which will help in cloning in a vector? Vector should possess something for which cloning is possible. You know what is cloning? We have already discussed, isn't it? So this is one vector, means in this one vector gene of interest is introduced. Now from this one plasmid you can find many plasmid has to form, isn't it? So many number of plasmids has to produce. That is called as what? Cloning, making multiple copies. So these copy number, the term what we use is what? Copy number. So if I am making only one plasmid, in that I am introducing my gene of interest, and if that plasmid, imagine if it is not replicating, if it is not dividing, it is not making its own copy, it is not going for cloning, then my process is an utter waste. So I have to have, this vector should have some characteristics. So what are the characteristics or what are the features? Okay, so the features when you are seeing, they should possess some characteristics, in which the main important thing is that they should produce some copy numbers, means they should produce many number of copies. Okay, so that I will be benefited, right? So any one copy numbers, if they are producing, many will be there. If one is not successful with the other one, we can go for the process, isn't it? So they have to have what? Good number of copy numbers, right? So when you are moving on to the features of a cloning vector, what are the features to facilitate cloning in a vector? There are three features. How many features are there? There are three features. What are the three features? One is origin of replication. So first one is what? Origin of replication. Second one is selectable marker. And the third one is cloning site or recognition site. Okay, so the first one is what? Origin of replication. Second one is selectable marker, and the third one is cloning sites or recognition sites, right? Now, origin of replication. What do you mean by origin of replication? This one we have already learned when I am saying lesser ORI site. What term I used? ORI, ORI site I used, isn't it? So here in this vector, you can find there is a site called as what? ORI site. So these are all different gene segments, like ORI is one gene segment, AMPR is another gene segment, PST1, PVU1, ECOR1, CLA1, HIND3, BAMH1, SAL1, PVU2. Okay, all these are what? Different DNA sequences coding for different characters. In that, the ORI sequence is very important. If this ORI sequence is present, then only this plasmid can produce many number of copies. Okay, they can go for replication from 1 to 2, 2 to 4, like that. For making copies, what is needed? This ORI sequence is needed. Origin of replication sequence is needed. Then only the DNA will be going for replication. From that, I will be getting many plasmids. PBr 322 I will be getting. If ORI is not there, the plasmid will not replicate. In that case, I am going for a waste. With one, it is not possible. So I want to make a number of copies, for which the important thing a vector should have is the site sequence, which is called as what? The ORI sequence, right? So this is regarding your ORI sequence. This is the sequence from where replication starts. Okay, I told you this is one DNA. So there should be a sequence, ORI sequence. So in that sequence only what is happening? Unwinding is taking place and replication is taking place. From 1 to 2 is formed. So this sequence is very important. If the sequence is present, then only replication will take place. Within the cell, you can find many number of copies of plasmids will be formed, right? And this sequence is also responsible for controlling what? The copy number. ORI sequence controls. Some plasmids will be making copies 10 times. Some may be making copies 30 times. Okay. So how many they are going to make? How many copy number? It is controlled by whom? The sequence. ORI sequences. In different bacteria, in different plasmids, the ORI sequences coding for different numbers. So that numbers we call it as what? The copy numbers. In some maybe they are producing 10 copies, in some maybe they are producing 30 copies, like that, right? So this is regarding what? Origin of replication, very important for forming clones, okay, for increasing the number of plasmids. So that is number one, what the vector should have. The second thing is that selectable marker. What do you mean by selectable marker? So I have already told you in that diagram what was the thing I have told you. So in this diagram you can find this is the plasmid, isn't it? Plasmid which is having the foreign DNA. Thereafter I have introduced this plasmid into the bacteria, the vector, isn't it? So from there you can find, because of ORI from 1, here 3 has been noted, like that many they are producing, right? So now listen very carefully. Selectable marker means what? Like this plasmid, when they are dividing, along with that the bacteria cell also divides, right? So if this bacteria cell is dividing and producing 4 bacterial cells, okay, in each they will have its own DNA. Yes, own DNA is present. Along with that, some plasmids will be present here, some will be present in this cell, some will be present in this cell, some will be present in this cell, right? Now listen carefully. In these all bacteria, this 1, 2, 3, 4 bacteria, I have to select which one is there carrying that rDNA, this foreign DNA, or this recombinant DNA, and which is not carrying a recombinant DNA. So out of this 4, it is not necessary that everyone who is replicating is carrying that particular gene. Sometimes it doesn't happen. That gene remains suppressed. It is not expressed. So in that case, they wouldn't have got that gene, isn't it? So I have to confirm whether out of these 4 cells, or the 4 cells are having my recombinant DNA. Recombinant means what? That foreign DNA, pest resistant, is present here. It is present in the second one. It is present in the third one. It is present in the fourth one. I have to confirm, isn't it? For confirming this thing, I need to have something like a marker. Okay, so what I should have? I have to have a marker, so that by seeing the marker I can understand this bacteria is having my rDNA. If the rDNA is not there, which means the marker is not there, then automatically the rDNA will also not be there. So there should be a selectable marker, right? Now moving on to the diagram, you can find what is the selectable marker. In this diagram you will understand. Selectable markers are usually resistant characters. Okay, so what are they? They are the resistant characters. So ampicillin is an antibiotic. What is ampicillin? Antibiotic. What is tetracycline? It is also an antibiotic. Now ampicillin resistant tetracycline resistant, which means if you are putting the bacteria, okay, all the cloned bacteria which I am getting, okay, they are having the plasmid, some are having 4, some are having 3, like that many plasmids are there. Now all these bacteria, if they have recombinant, means rDNA is present, which means that they will be expressing the character. They are resistant to ampicillin. They are resistant to tetracycline. How I can confirm? I will take a tray. Okay. In that I will be adding ampicillin. What I am adding? Ampicillin. After adding this ampicillin, what I am going to do? I am going to take this 4 bacterial cell and put it in this tray. Okay. Now what will happen? If this bacteria are having this plasmid, which is having my gene of interest, if that plasmid is present, then automatically what is happening? They will be resistant to what? They will be resistant to your they will be resistant to what? They will be resistant to ampicillin, right? So this ampicillin medium, isn't it? So they will be resistant to what? The ampicillin, which means these 4 bacteria will survive. They will all survive. Now imagine, in case I am not taking, means out of this 4, 1 is not surviving, which means that this cell, this bacterial cell is not surviving. They are dying. What is happening to them? They are dying, right? So this has died. Now, which means that in that what is not present? That plasmid is not present. It is not expressed. Okay. In that case, that bacteria is not a transformant. They are not carrying the rDNA. So in order to confirm whether all the bacteria what I have taken, which is carrying the rDNA and which one is not carrying the rDNA, I want to have some marking on their bodies, isn't it? So the marking is like a selectable marker, right? Understood? So you can see here, it is a sequence in DNA which helps in selecting transformants. Transformants mean what? Which is having the rDNA. rDNA I have told you, rDNA is what? The one which is having the foreign DNA, we call it as what? The rDNA. So this rDNA, if it is having or not, how will I confirm? I will confirm with the help of selectable marker. So with the help of selectable marker, I am going to confirm. Okay. So the transformant which is containing rDNA and eliminating the non-transformant, the bacterial cell which is not having the rDNA, I should not take for my study. I have to eliminate them. I have to take only the clones which are containing the rDNA. So how can I confirm? With the help of what? The selectable marker. So normally the genes which is encoding resistance to antibiotics like ampicillin, tetracycline, kanamycin, all these are acting like what? Selectable markers. So here, which are the selectable markers? I have already told you which are the selectable markers. The selectable markers are ampicillin resistant and tetracycline resistant. So when I am taking a bacteria, so I have introduced the rDNA into this bacteria is producing innumerable number of bacteria. All these bacteria is containing some amount of plasmid. Okay, all are having what? Some amount of plasmid. Now if this I am putting in an ampicillin medium, if all these bacteria are surviving, means what? They are resistant to antibiotic. So rDNA is present. If they are not surviving, they are dying, means this plasmid is not present in them, which means that they are not containing the rDNA. They are non-transformants. I have to eliminate those bacteria. Like for example, if this is not containing, I will take this colony out. If this is not containing, I will take it out. Okay. Only this one I will select because they are resistant to antibiotic. So they are surviving. These guys are dying, which means they are not resistant to that antibiotic. So that is acting as what? The selectable markers. Okay. So what are the selectable markers in this? I told you ampicillin resistant and your tetracycline resistant, right? Now, normal E. coli cells, when you are seeing, they do not have this special E. coli cells which is acting like vectors. What I am selecting, they have this ampicillin resistant, tetracycline resistant. One of the example is what? PBr 322. Others, they don't have this other E. coli, they do not have this ampicillin resistant and tetracycline resistant characters. So the vector what I am selecting should have what? A selectable marker. And what is the selectable marker? It is the resistant character. So what are the resistant character? They are resistant to certain antibiotics. So what are the antibiotics? It is tetracycline, ampicillin, kanamycin. The transformant will be growing in that medium containing these antibiotics. The non-transformant will not grow because they are not resistant to, say they cannot live in that medium. Understood? So that is the role of what? The selectable marker. Hope it's clear. Now moving on to the next important thing, what a vector should have is a cloning site or the recognition site. Okay. So what is a cloning site and a recognition site? Say listen. So one vector, this is a vector, means they should have a cloning site. So what is cloning site? Cloning site is the site, it should have only one cloning site. That is very important. Okay. So a vector should have a very few, preferably single cloning site. Like for example, if I am drawing, this is the plasmid. Okay. So in this plasmid, there should be only one recognition site. So this is the recognition site, imagine. Why this recognition site should be one? It should not be many, because if I am adding a restriction enzyme, what I am going to add? Restriction enzyme. If this restriction enzyme, if this vector is having many cloning sites for this restriction enzyme, then what will happen? They will go and make a cut in many places, isn't it? So many places will create, the sticky ends will be created in many places. Now it will lead to confusion. In which one I will introduce my DNA, and again with DNA ligase I have to paste this one, other segment, everything I have to paste, isn't it? So in order to avoid that, I will be selecting a vector which is having very few. Sometimes I will prefer having only one recognition site, which means if I add a restriction enzyme, correctly it will go and cut at particular site. They will not go and cut in any other site excepting that one single site, which means it was easy for me, because here if the sticky end is created, my foreign DNA, where I can put it over here and then ligate it. Okay, I need not ligate in some other places. This place on one cut, so there I am putting it and I am ligating it. I am getting my recombinant rDNA. So gene of interest is ligated where? In this cloning site it is gone. The foreign DNA is being introduced. Okay. If many recognition sites is there, I told you what is the problem. Many cut ends will be produced, sticky ends will be produced. One foreign DNA I have to introduce in one sticky end, others and all I have to paste it again. It's tiresome. Okay. So preferably there should be only one cloning site, so that correctly their restriction enzyme when I am adding, they will cut there, sticky end will be created, my foreign DNA will be inserted into it, and it will be pasted with the help of what? The DNA ligase. Okay. So presence of more than one recognition sites within the vector will generate several fragments, I told you. If more are there, many sticky ends, many fragments will be there. Ligation of the foreign DNA is carried out at the restriction site present in one of the two antibiotic resistant gene. Now listen, it is very important. Now in this you see, usually the ligation means what? Restriction site, recognition site. What I am going to do is that this PVR 322 is having two selectable markers. One is ampicillin resistant and another one is tetracycline resistant. Ampicillin resistant, ampicillin is an antibiotic, tetracycline resistant, tetracycline is an antibiotic. So when I am introducing my foreign DNA in this normal plasmid E. coli, which is having both ampicillin resistant and tetracycline resistant, I will select one of the marker as a cloning site. Okay. So one of the marker is what? Either ampicillin or tetracycline. For example, I am selecting BAM-HI, this tetracycline resistant gene, that is the selectable marker, that I am suppressing it, I am taking it out. So when I am adding a restriction enzyme, this tetracycline resistant gene sequence is cut. Okay, it is removed. Now what I am going to do? I am going to introduce the foreign DNA, isn't it? The gene of interest, like pest resistant gene is introduced into this. Okay. So what is removed? Tetracycline resistant is removed with the help of that restriction enzyme. and that sequence is removed, and in that sequence I am going to introduce my foreign DNA, that is DNA which is coding for pest resistant, right. Now listen carefully. This one, after introducing, I have got my vector, the plasmid is ready. Now this plasmid is put into the bacteria, okay. Now this bacteria will go multiply into many bacteria. Within them also the plasmids will multiply. Like that many bacteria will be formed. Many are having many plasmids, right. Now I have to select from this which one is a recombinant and which one is a non-recombinant, or which one is a transformant and other one is non-transformant. How can I select? I can easily select by, first I will put all these bacteria in a medium, okay. So I am taking all these bacteria, small bacteria I will draw. So small bacteria I have taken, and first I am putting it in ampicillin resistant tray. Where? So ampicillin medium. So here a tray with ampicillin. Now all these transformant as well as non-transformant will grow, because ampicillin resistant is present in transformant as well as ampicillin resistant is present in all these things, okay. But tetracycline, next I am going to take all these bacteria. I am seeing that everyone is growing very nicely, so no problem. Everyone is having the gene of interest, no problem I think. But I have to confirm, isn't it, which and all is not having. So what I can do? Afterwards, what I will do is that I will remove this, and then I will add a tray. All these bacteria I will collect, and I will add in a tray containing tetracycline. What I am going to add? I am adding tetracycline in a tray. In that I am going to introduce all these bacteria. From ampicillin medium I will take and put it in the tetracycline. Now listen very carefully. Tetracycline resistant factor is removed, isn't it? Tetracycline, it was resistant. First they can grow in tetracycline, but I have removed the tetracycline resistant, and in that place only what I have done, I have introduced my foreign DNA. Now this rDNA is not resistant to tetracycline. They cannot resist what? The tetracycline. Why it cannot resist? Because tetracycline resistant character is removed. The gene is removed. Now if you are putting them in the tetracycline, they will die, which means they cannot survive if there is tetracycline, because the tetracycline resistant gene is removed. In that place what I have introduced? My foreign gene, isn't it? So if I am putting these bacteria in the tetracycline, what will happen? The bacteria which are resisting and growing in that medium, which means that the recombinant is not present. It is a not recombinant. What I have done, it is not that. Some I see that they are starting to die, their colonies are starting to die, which means they are recombinant, because that in that only I have introduced my foreign DNA, isn't it? So they cannot survive means what? They are slowly dying out, their colonies are slowly dying out, which means that tetracycline resistant gene is suppressed and my gene is possibly expressed, isn't it? So I will be selecting those colonies which are dying fastly and transfer them, which from that I will be getting what? All the bacteria which are having what? The recombinant pest resistant gene is present. So for selecting this one, cloning site is playing a major role. This one, those who have not understood, once again I will tell you, it's a bit difficult to understand, anyhow I understand, okay. Cloning sites. So usually cloning sites, out of the two resistant character in PBR322, I select one, okay. So in this, when I am adding a restriction enzyme, what is happening is that the tetracycline resistant gene, from there you can find what is created. A sticky end is created and that segment is removed. In that only I am going to introduce what? My foreign gene. So what is the foreign gene? The foreign gene is the pest resistant gene is introduced over here. Now this rDNA what I have made, this rDNA is not resistant to tetracycline, which means if I put this plasmid into tetracycline, they will die. That bacteria will die. Okay. So why it will die? Because that resistant is removed and the foreign gene is introduced. Now this rDNA is introduced into the bacteria and the bacteria is allowed to multiply. Okay, the plasmid is allowed to multiply, the bacteria is allowed to multiply. Like that, what I am getting many innumerable numbers of bacteria. Okay, now from these bacteria I have to select which one is carrying my foreign genes. Only that I have to select. Which is not carrying my foreign genes, I don't want. So first I will put all of them in which one? Ampicillin solution. When I am putting all these bacteria in ampicillin containing solution, what is happening? All the bacteria are surviving, which means all the bacteria are resistant to ampicillin, they are surviving. But these bacteria are collected and then they are put in which one? They are put in tetracycline, where they are putting in the tetracycline. Now when they are putting in the tetracycline, some are surviving, some are not surviving. Surviving means what? The tetracycline gene is expressed, which means my foreign DNA is not inserted properly there. Okay. So which is surviving is not a recombinant. It is not an rDNA. Which is not surviving, which means that there my foreign gene has taken, that's why it is not surviving, because the resistant factor is lost. Okay. So there you can find what is happening. They can not survive, the recombinants cannot survive, and the non-recombinants can survive in tetracycline resistant. So these characters a cloning vector should have. So what are the characters? I have told you. First is that they should have origin of replication ori site, so that they can make innumerable number of copies by replication. Second is that they should have a selectable marker. If only marker is present, like resistant characters are present, then only I can understand which is a recombinant and which is not a recombinant. Okay. And the third one is what? They should have a cloning site where I can introduce my foreign DNA. Okay. Usually the sites are what? The resistant sites are used as what? The cloning sites. Now next what we are going to discuss is, we are going to discuss about vectors for cloning genes in plants and animals. Okay. So what is the next topic? Vectors for cloning genes in plants and animals. Now when you are seeing what is this vectors for cloning in plants and animals. So vectors, which vector we use to put into plants and put into animals. Okay. The vector for plant usually we use is Agrobacterium tumefacium. It is a pathogen. It is a pathogenic thing. The bacteria is a bacteria. Okay, it is a pathogenic bacteria. So what is this characteristics of this Agrobacterium tumefacium is that it is a bacteria and infects a number of dicot plants. Whatever dicot plants it is, Agrobacterium tumefacium, they prefer to go and attack them. Okay, they prefer to go stay on them. Thereafter they induce tumor in those plants. Tumor means, you know, the cells starts to divide in an uncontrolled manner. So that is the role of what? This Agrobacterium tumefacium. So Agrobacterium tumefacium is a bacteria. They are pathogenic. Pathogenic means what? They are disease causing. So if this is put into the bacteria, so this bacteria is put into the plant in that you can find they induce what? Tumor. What they induce? Tumor. Okay. So it is a pathogen of several dicot plants and is able to deliver a piece of DNA. What is called? That DNA piece we call it as what? It is called as tDNA. What is the DNA's name? tDNA. And this tDNA is converting the normal plant cells into tumor cells. Okay. So that is the function of what? This Agrobacterium tumefacium. It is not a good thing, isn't it? It is going pathogenic, attacking the plant, and it is inducing tumor. But it is not a good thing. But what we are going to do? I am going to take out this Agrobacterium tumefacium and use it as a vector. How I can use it as a vector? I have to introduce my gene of interest, like disease resistant variety or pest resistant variety. Where I have to introduce? I have to introduce in that place where it is producing some chemicals which is toxic to the plant, isn't it? So that chemical, the poison, is responsible for causing the diseases. Pathogenic means what? It is causing diseases. Tumor inducing. Okay, so this tumor inducing TI plasmid, now what I am doing? I am modifying it. How I am modifying it? That region which is pathogenic is removed from the vector, and in that what I am introducing? I am introducing my foreign DNA. Okay, so which one is removed? Imagine this is the plasmid present in Agrobacterium tumefacium bacteria. Now this region is the region. So this is the region which is causing what? This is causing pathogenic. Pathogenic means what? Causing the diseases in the plant. Okay, tumor or disease is caused in the plant because of this sequence. So what I am doing here in biotechnology, I am removing this sequence. I am taking out this sequence, and in that I am introducing what? I am introducing my foreign DNA. So what is a foreign DNA? Pest resistant is introduced there. Okay. Now what will happen? This Agrobacterium tumefacium, I am putting it in the Agrobacterium tumefacium, this plasmid modified one, rDNA, I am putting in where? In the Agrobacterium tumefacium, and this Agrobacterium tumefacium is going and attacking the plants. When they are going and attacking the plants, which one is introduced? This plasmid is also introduced into the plant. Along with the plasmid, what is present? My foreign DNA, isn't it? So pathogenic, is it pathogenic? No, that is removed. Now it is what? It is helpful to me because it is introducing the DNA sequence, the foreign DNA, which is coding for what? Pest resistant. Like that, I can develop many different varieties of plants resistant to drought, resistant to pest, with the help of what? This Agrobacterium tumefacium. By removing the pathogenic sequences and introducing my gene of interest into that, I can develop varieties of plants and animals which are resistant to several, not animals, plants which are resistant to several characters. Okay. So this is regarding your Agrobacterium tumefacium. The same function, okay, Agrobacterium tumefacium in plants, they cannot attack us. In animals, we cannot introduce this Agrobacterium tumefacium. They are specific to plants. But in case of animals, we are using another one vector. What is the vector we are using? We are using a virus, a group of virus called as what? Retroviruses. In that, the virus property, the poison controlling property is removed, and in that my gene of interest is introduced, and then it is put into the animal. Okay. So when it is putting into the animal, this virus is no longer acting like a virus, because the virus which is responsible for causing any diseases, causing some toxic substances, is disarmed. Disarmed means what? I am taking out its arm, which means I am taking out its property of causing something harmful. It is being taken out, and in that sequence, what I am introducing? I am introducing my gene of interest. So in animals, we are using what? The retroviruses, and in case of plants, what we are using? We are using Agrobacterium tumefacium as a good vector. So vector for plants, if you are asking, it will be Agrobacterium tumefacium. Why? Because they contain TI plasmid. Now this in this TI plasmid, tumor inducing plasmid. So in that, I am removing the segment which is causing diseases, and in that I am introducing my gene of interest and delivering the gene into the plant through a vector, Agrobacterium tumefacium. The same in animals, we are using what? The retroviruses. Hope it's clear. Next topic what we are going to discuss is insertional inactivation. Again, it is coming under vector only, the topic insertional inactivation. Okay.